Thank you very much, Nicola. Okay, so thank you all for having me. And yeah, as you said, I'm going to be talking today about gene drives for genetic control of the malaria mosquito. I think I'll jump straight in. So I usually like to start my talks by just highlighting the problem that we're dealing with. I'm sure many of you will know this already, but the mosquito is the world's deadliest animal. And that's actually primarily through the transmission of just one disease, that's malaria, which claims well over 450,000 lives each year, and predominantly in children under the age of 5. And when we think about where malaria is, it's an insect species which is actually widely spread throughout the tropics. And as you can see on this map, that gives you an idea of where you find Anopheles mosquitoes that transmit malaria. You find them in Central and South America. You find them in Africa and sub-Saharan Africa, and you find them throughout the East. But when we talk about the population at risk, it's not actually just anywhere where you find malaria that you have a real problem of malaria. It's actually just these regions in red, which are almost entirely in sub-Saharan Africa and the Indian subcontinent. And that's actually because of this combination of the vectors that you have, the particular mosquito species in those regions, and the types of parasites that you find in those regions. And the complementarity between them leads to very, very high rates of malaria transmission. So we have a problem that is predominantly affecting the underdeveloped world. So how do we currently fight malaria? Well, we have strategies where we can target the parasite. And for this, we have antimalarial drugs. And there's now new excitement about vaccines. There are some vaccines which have already begun to be used just beyond phase 3 trials. They're not incredibly effective, but there is excitement that new vaccines, and there's some discussion of mRNA vaccines or whole sporozoite inactivated vaccines. These could be quite an effective tool against malaria. But right now, we don't have something that is suitable for mass use in the regions where people are most affected. But what I can say is that we do have very effective ways of controlling malaria at the moment based on targeting the mosquito vector. And to do this, there are several strategies, and almost all of them rely upon the use of insecticides. So for this, it will include indoor residual spraying. It would include removing habitats and biological control, insecticide-treated bed nets. And there are some strategies based on fumigation, which in some contexts may be effective, but for malaria control, this is generally not an effective strategy. What I would say is that there are 2 interventions which have been absolutely critical to the incredible successes we've had in malaria control over the last 15 years. And that's indoor residual spraying and insecticide-treated bed nets. And these 2 interventions alone, it's thought that they're responsible for more than 75% of the reduction in malaria transmission that we've seen over the last 15 years. And that's a reduction from around 1 million deaths per year in the year 2000 to nearer 450,000 deaths per year now. So when we talk about the mosquito, I want to be a bit specific here, because there are actually 3,500 species of mosquito. And there are actually very few of these that transmit disease to humans. And it's believed that there are just 3 main species complexes that transmit 95% of malaria. And I would like to update the slide a little bit, because I say Anopheles gambiae, Anopheles colutzii, and Anopheles arabiensis. But actually, I think it's worth putting in Anopheles funestus now. And we have these species complexes, the Anopheles gambiae species complex, which includes Anopheles colutzii. We have the Anopheles arabiensis species complex, and we have the Anopheles funestus species complex. And those 3 together are responsible for the vast majority of malaria transmission worldwide. And again, I don't want to be cruel to all of those mosquitoes, because they're not all bad. It's only the females. And that's because male mosquitoes, they do not bite and they do not transmit disease. It's only the females that bite. And they do this because they take blood and they use that blood for the proteins to make eggs. So how can we specifically target the most dangerous mosquito species? And what I would say is that there are a number of different strategies being developed, and many of these can be effective, and that we are unlikely to reach malaria elimination without using a range of different tools. But what I'm most excited by are those that use the mosquito itself. And one of these is called the sterile insect technique. And the basic idea is that you would rear mosquitoes in the laboratory, and you would remove the female mosquitoes, those that are capable of biting. And what you're left with is the males, and you would sterilize those males. And how you sterilize them is one of the tricky parts of this process. But once you have these sterile males, the idea is that you'd release these en masse into the local environment so that the environment is flooded with sterile males, such that any wild female is more likely to mate with one of your sterile males than another wild type male. And because mosquitoes are species that rarely mate more than once, if a female mosquito mates to your sterile male, it is a dead end and will result in that female being effectively sterilized. So if you keep releasing these males at a large excessive number each generation, then you can have a reduction in the total number of mosquitoes in that population through this sterile insect technique. The strategy can be incredibly effective, but it relies on a large number of insects being reared and released, sterilized and fit, and actively mating with that population in the wild each generation. It's a long-term strategy, and it's quite an expensive strategy depending on the species and depending on the situation. So when we think about malaria control in sub-Saharan Africa, what we need to consider is the infrastructure that you have. And it's unlikely that we will have the rearing capabilities and, in fact, the money that's necessary to carry out a sterile insect technique program like this for malaria control in sub-Saharan Africa, when we're already falling far short of the funding that's needed just to keep up the current level of control. But the idea is that if you can bring the mosquito population to such a low level, you can reach malaria elimination. And that doesn't mean eradicating the mosquito. It means bringing the number of mosquitoes low enough that the cycle of transmission is broken. The malaria parasite must always exist in a mosquito or in a human. And if you can break that cycle, you can actually get to malaria elimination, either locally or hopefully on a regional level. But as I said, right now, the release on this scale is not possible in sub-Saharan Africa. So as a way to move forwards, something that I became interested in is genetic modification. And what you're seeing here on the left is that's a mosquito embryo. And that's a video that I took many years ago of me injecting it. And so you see this egg, this little banana-shaped thing, that's needle going in, and we can inject in DNA, and we can make genetically modified mosquitoes, and we can maybe use that to enhance this technique of the sterile insect technique. And this is something that works, and we can use genetic modification to do interesting things. We could use it to immunize the mosquito against malaria transmission, or we could use it to reduce the total size of that mosquito population, something similar to the sterile insect technique. And both of these approaches are very, very interesting, and they could be complementary. But what I focused on is population suppression. So I'm focused on ways to reduce the total number of mosquitoes using genetically modified mosquitoes as a means to do that. So here you see some mosquito larvae swimming around, and these have all been engineered to express the red fluorescent protein. So when I turn off the lights and I turn on my fluorescent microscope, you can see them glowing in their eyes and down the body. And this is a genetic modification we've introduced that lets us see them visually through this red fluorescent protein. But we still have this problem. We can make genetically engineered mosquitoes, and we can do interesting things, and we can perhaps immunize them against malaria, or we can perhaps make them sterile. But how can you introduce a genetic modification to an entire population in the wild? And when we think about sub-Saharan Africa, if we're going to rely on just releasing large numbers of these mosquitoes, it's not possible. It's not possible on that scale. So how do we get over it? So gene drive is a relatively new concept towards something that's been achievable in a lab setting. And what I want to show you is just the basics of gene drive and how the inheritance of a gene drive is different from a normal genetic modification. So for this figure and for my sort of remaining slides, I use a theme. So blue mosquitoes are a wild type or unmodified mosquito, And if it's red, that means it's been modified to contain a gene drive, or in this case, a non-gene drive as well. So without a gene drive, if two mosquitoes mate and your genetically modified mosquito is heterozygous, you would expect a 50-50 transmission of that gene. And over time, you would expect that that transmission stays the same, such that your genetic modification would not spread. And more likely than not, we know that when we introduce mutations into an organism, very often they're not as healthy as they were before that modification. So the most likely outcome is that a genetic modification that we make and we release, that will get lost over time. And it will only stay at a maximum frequency of the original frequency we release it at. But what a gene drive does is a gene drive can bias its own inheritance. So a gene drive can make sure that it gets inherited by more than 50% of the offspring, and perhaps even 100% of the offspring, even when it's heterozygous. And this process is self-sustaining. So with each generation, every time your transgenic mosquito containing a gene drive mates with a wild one, it can double in frequency in an ideal scenario. But you don't need that doubling, even if there's a small bias in transmission, even if it's 55% get the gene drive and 45 don't. That will cause it to start spreading in a population over time. And that spread is very rapid. In fact, it takes about 20 to 30 generations for a gene drive to become fixed in a population, even when you release it at an incredibly low initial frequency. And you can use this to spread anything you want, really. You could use it to spread a trait which makes the mosquitoes resistant to the malaria parasite. You could make them susceptible to the insecticides. Right now, we have a problem with insecticide resistance that is spreading through natural populations. You could engineer mosquitoes to remove that insecticide resistance and enhance the power of traditional insecticide-based approaches. You could maybe try and change their behavior so they don't want to bite humans. Or what I've been interested in is introducing what's called a population suppressor, something that will spread through the population, but reduce the potential of that mosquito population to exist in large numbers. So this seems like a bit of a paradox, because what I'm going to say is that I can spread a gene that makes mosquitoes infertile, but it spreads through reproduction. So how can a gene spread through reproduction if the mosquitoes are sterile in the first place? So we have just two rules that you need to follow for this to make sense. And the first rule is that the gene drive must not cause infertility when it's inherited from one parent. And the second rule is that it only makes female mosquitoes infertile. So only females will be infertile, and only if they inherit it from both parents, but not from one. So let's see what happens. Your gene drive mosquito mates with a wild one, and of course you get a bias in the transmission. So that mosquito is going to continue mating with more wild mosquitoes, and you're going to continue to get this bias, and you're going to see a spread in the population. But nothing is sterile, because at each step, our gene drive mosquito, one of its parents was always wild type. So because of that, it's unaffected by the gene drive. But when two gene drive mosquitoes mate to each other, they're going to produce these homozygous progeny, of which the females will be sterile, but the males will still be fertile. And so too will any gene drive mosquitoes that had a wild type parent. So you have a situation where when the gene drive is rare, it's mostly going to meet non-gene drive mosquitoes. It's going to meet wild mosquitoes. And because of that, it's going to spread through the population. But as the gene drive starts to become very, very common, they will start meeting each other. And once they meet each other, they'll start producing progeny whereby the females are sterile and the males are still fertile and still able to continue spreading that gene drive. So the effect of this is that if you were to release just a small number of gene drive mosquitoes on day one, you don't need to re-release them. That gene drive will spread on its own. It will bias its own transmission and drive itself through the population. So that's what we see in this red line. And as it's spreading, you'll see a reduction in the total number of mosquitoes as more and more of the females are made sterile. And if you reach a low enough level of mosquitoes, you can actually get to malaria elimination. So this strategy is incredibly quick. And I told you it can work in 20 to 30 generations. And a mosquito reproduces once every 3 weeks or so. So 20 to 30 generations is actually just a couple of years. Now, of course, this is an ideal scenario, and this is just assuming everything works perfectly, everything mates perfectly, and there's no strange dynamics going on. But in an ideal situation, a gene drive could spread through an entire population, modify it, and suppress it within a couple of years. So how do we build them? So the idea was actually developed in 2003 by Professor Austin Burt at Imperial College London, and he developed this idea called the driving endonuclease gene. And the idea was based on technologies before CRISPR existed, but CRISPR actually fits into that in the same way. And it doesn't make any real difference. So for the sake of this talk, I'm going to focus only on CRISPR. But the idea is that a gene drive contains just two things. It contains a Cas9 and a guide RNA. And so in red, in this diagram, I'm showing you the gene drive itself, and that's just the genes for Cas9 and guide RNA. And what we've done is we've programmed this Cas9 and guide RNA to recognize a gene that females need to reproduce. And not only have we programmed it to recognize this gene, we've actually inserted the gene drive inside that female fertility gene, just as I've shown in this diagram. So if you were to express Cas9 and guide RNA from this chromosome on the right, it's going to come along and find its target site inside this yellow gene, inside the female fertility gene, and it's going to cut it. And once that chromosome is cut, it needs to repair. And the best way to repair is by using your unbroken partner chromosome as a template. So this is homology-directed repair, but in this context, we call it homing. And this is really the driver of gene drive. This is the motor. This is how it works. And this process continues going on every time a gene drive chromosome sees a non-gene drive chromosome. It's going to cut it and copy the gene drive inside. So I want to give you a quick overview of the whole strategy as I presented it. So we have a promoter which will express Cas9 in the germline of our mosquitoes, and a promoter which will express guide RNA. And together, the Cas9 and guide RNA will come along and they will cut a gene on an unmodified chromosome. And in doing so, by homologous recombination, the genes for Cas9 and guide RNA will get copied inside. So that's a gene drive copying from one chromosome to another. You can think of it as converting a heterozygote into a homozygote. Now, the way this works is that we program this to happen only in the germline. And this is a slightly complicated part of this process. And I'd say if you don't follow it and I haven't explained it well, I wouldn't worry too much. But if a mosquito is homozygous in its entire body, it's going to be sterile as a female. But we get over this problem because we program this reaction, this homing reaction, to happen only in the germline. So the rest of the mosquito is heterozygous. The rest of the mosquito still contains that female fertility gene. And because it contains that female fertility gene, heterozygous gene-drive mosquitoes are fertile, even though their germline has been converted to homozygosity. So it's only when you inherit the gene drive from two gene drive parents are you fully homozygous in the somatic tissue and the germline. So every time we have a fertile female, it must mean that her somatic tissue is heterozygous or wild type. And every time we have a sterile female, it's because her somatic tissue is homozygous. And that will only happen if you inherit the gene drive from two gene drive parents. And of course, the idea is that we release a small number of these, it spreads through the population and causes a widespread suppression. So how do we build these? So initially what we did is when I started my PhD, I was set about finding the right genes for us to target that would make female mosquitoes infertile. So to do that, we used CRISPR and we targeted several of these genes and we just inserted a marker gene, GFP, inside. And then we checked in heterozygous females to see if they were fertile. And we want the heterozygotes to be fertile. We want them to be fertile when just one copy of the gene is broken. But when two copies of the gene are broken, those females were sterile. So we checked the number of larvae in females carrying mutation to one copy of the gene, and they look just like wild type. When we mutated both copies of the gene for these three different genes, 11377, 5958, and 7280. Those 3 genes resulted in complete sterility of females with a homozygous disruption, and it didn't affect males at all. So the next step is, okay, can we convert this into a gene drive? So to do that, we used a little trick called recombinase mediated cassette exchange, where you can swap that GFP for the instructions for a gene drive. So that's the Cas9 and the guide RNA. And the way that we know this trick has worked is that we swap the GFP marker for an RFP marker. So the mosquitoes will convert from being green to being red under a fluorescent microscope. So the next obvious question is, does it work? Do these gene drives spread? So we have a very, very simple experiment to test this. We take a mosquito, male or female, that has 1 copy of the gene drive, and we simply mate it to a wild one, and we count the offspring, and we say how many of their offspring are shining bright red, how many of them inherited that gene drive. And if it's 50%, the gene drive doesn't work. And if it's more than 50%, the gene drive is working. It's copying from one chromosome to the other and biasing its own inheritance. So we did that looking at crosses of males and females for each of these 3 different gene drives. And in each case, we get very, very high rates of transmission. So these are ranging from around 93% to 100% of the progeny containing the gene drive. So that's very, very effective. So then what we did was we decided to do a cage release experiment where we take our gene drive mosquitoes, we throw them into a cage, and we track them over time. The expectation being that they're going to spread. And what we saw is that when we released a 50% initial frequency, the gene drive spread very quickly up to 80% in 2 replicate trials. But the prediction was that this would reach 100% within about 12 to 15 generations. And when we followed that gene drive for more than 1 year, we found that actually it got lost. So this is exactly the opposite of what you'd expect. The gene drive is supposed to spread and supposed to spread all the way to every mosquito in that population. And instead it spread for a while and then it got removed from the population. So we figured this is some kind of molecular resistance. And in fact, if you go back to Austin Burt's original paper from 2003, this is something that was predicted to happen and something that we expected. So how might this happen? So your gene drive is programmed to recognize and cut some essential gene. But of course, when you do CRISPR, sometimes you introduce mutations, and those mutations might be resistant to further cutting. So you can imagine that we program our guide RNA to recognize a 20 base pair region of the genome. And if there's 1 or 2 changes to that 20 base pair region, the gene drive can no longer recognize it and no longer cut. So we would call that a resistant mutation. But it's a little bit more complicated than that, which I'll get onto later. And these resistant mutations can be generated by CRISPR through the end joining pathway. And this is something that's very common and very well studied. But they could also be naturally occurring. We know that the mosquito genome is very diverse. And just like humans have differences in our DNA, mosquitoes have differences between individuals. And it could be that some individuals naturally have a version of this gene which cannot be recognized by our Cas9 and guide RNA. So we'd call this naturally occurring resistance. But really, it's just something which is just a naturally occurring genetic variation. And the last thing to consider is that any resistance that you do get is going to have a massive selective advantage. Because if we're trying to sterilize female mosquitoes, then if your mosquito is not sterilized, there's a massive selective pressure for whatever that resistant mutation is. And that the stronger the impact of the gene drive, the more severely it impacts the fitness of your mosquito, the more strongly you're going to select for resistance. So not all resistant mutations are actually truly resistant to a gene drive. And that depends on what type of gene drive you're building. But if you're building the suppression drives, we're targeting essential genes. So if you have a mutation in that essential gene that breaks it, then your mosquito might be resistant to the gene drive copying, but it's going to be sterile. So what I mean is, this essential gene that I have in blue, if this is essential for making eggs, and there's been a mutation that stops the gene drive from recognizing it, that might not matter if it turns out that the egg producing gene is no longer functional. Because how can that spread in a population? It won't. So we have these non-resistant mutations that might be able to stop the gene drive from spreading, but they can't actually stop the gene drive in the grander scheme of things, because if that were to spread on its own, mosquitoes would be sterile. So what's really a problem is if you have mutations where the gene has changed, but it's still functional. And it's changed in a way that the Cas9 and guide RNA can no longer recognize it, but it doesn't impact upon the mosquito's own fitness. So this is the real problem. And we call these R1 resistant mutations. The non-resistant mutations, we call them R2. So a little bit more on resistance and end joining. So we wanted to understand what frequency does this occur at, and where is it occurring, and where do we need to focus our resources to try and solve the problem of resistance. So the first thing we did was we looked at how often these mutations are generated in the germline of female mosquitoes versus males. So in both instances, we see the gene drive copies about 99% of the time. But that remaining 1%, we wanted to know what happens to them. And the remaining 1%, it turns out in a female, most of them will contain a resistant mutation. Whereas in the male, they don't have as many containing a resistant mutation, but we did see something really interesting. So females, if they did not manage to copy their gene drive into that chromosome, they will almost certainly have generated a mutation inside instead. But most of the progeny will contain the same 1 or 2 mutations. But in a male, whilst the rate of end joining, the rate of mutations is lower, the diversity of mutations is much greater. So we found a wide range of different mutations being generated in the male germline. And because only some mutations will be resistant, it's like throwing the dice. A female is going to generate many offspring containing the same mutation, but that mutation may or may not be resistant. Whereas a male will produce just 1 or 2 mosquitoes containing a mutation, but they contain lots of different mutations. So the chance that you find one which is truly resistant is higher from a male because of the diversity of different mutations that they produce. So females, it was about 85% of their non-gene drive progeny that contain a mutation. And for males, it's around 35%. Now, this is something that we published this year. So these rates, even though they're not great, they're not high enough to explain how quickly our gene drive failed when we released it in caged experiments. So we wanted to ask the question, when is resistance really generated? Because that isn't enough. That low rate, that 0.5% is not high enough to have caused our gene drive to have failed after 6-7 generations. So we came up with an experiment, and I thought this was a very fun idea, and I think that we learned a lot from this. So I want to go through it quite simply, but in detail. Imagine we have a mosquito that contains on one chromosome, it contains the gene drive in red, and on the other chromosome, it contains a resistant mutation. So this is something that cannot be cut by the gene drive. So if we cross this mosquito to a wild type, you would expect 50% will inherit the gene drive, and the other 50% will inherit this resistant mutation. So these individuals on the right, if we were to sequence their chromosomes, you should see 2 things. You should see the resistant mutation, and you should see the wild type chromosome, but nothing else, surely. Because they do not contain the Cas9 and the guide RNA. The Cas9 and the guide RNA are present only on the gene drive, so only these individuals on the left should be having some evidence of CRISPR. Those on the right, they should have nothing. But actually, we decided to look at these because we had a hypothesis that something might be happening. And our hypothesis was that the parent of that mosquito inherits Cas9 and guide RNA from the parent gives Cas9 and guide RNA to its offspring through this process called deposition. So what I mean is that when the female is generating her eggs and she's producing Cas9 and guide RNA, she can package the Cas9 and guide RNA into those eggs, even if those eggs do not contain the genes to produce their own Cas9 and guide RNA. And so when we sequenced these individuals that do not contain the gene drive, but their parent did, we found that actually they carried tons of mutations. And this was only happening when the parent was a female. So the females are packaging their eggs full of Cas9 and guide RNA, but the males are not packaging their sperm with Cas9 and guide RNA. And this rate of mutation was happening at such a high frequency that this could explain pretty much all of the resistance we found in our experiments. And we also confirmed that this is heritable. So these mutations don't just happen randomly in the somatic tissue, they happen in the germline, they get inherited, and they are a massive problem for gene drive. So we then set about developing new systems to express Cas9 and Cas9. And as you'll see on the top, we have this thing that says Vas2. So Vasa is a promoter that we use to express Cas9. And this is what's causing the mosquito to make tons of Cas9 and to package it into those female embryos. So we explored other promoters to express Cas9 that would hopefully mitigate that process so that you don't end up with a whole load of this enzyme being packaged into the embryos. And these two promoters are called nanos, or NOS, as we shorten it, or zero population growth, we shorten it to ZPEG, CPG. And what you see is that for ZPG, we had no evidence of any mutations happening in the progeny. So it seems that we've solved this issue, and no longer do you get Cas9 and guide RNA going into the offspring and mutating them. So we decided to take a look at these individuals with the ZPEG version of the gene drive and release them into a cage and see if they perform better. So the original gene drive, using this terrible Vasa promoter, it was able to spread for a few generations, and then it went down and failed. And when we sequenced what types of mutations we had, we saw that very early on, we had a whole load of different mutations being generated, including resistant mutations, truly resistant R1 mutations, which are in orange, and these non-resistant mutations in blue. And at the beginning, when the gene drive is first spreading, you have all these different mutations being generated, but there's not much selection pressure. But as the gene drive is spreading through the population, you see a stratification where all of the mutations that are R2, non-resistant, they get removed, and all the mutations that are truly resistant, the R1s, they get selected. And you see that very, very clearly through this shift in the orange going from low frequency to high frequency, but the blues, they go from high frequency to low frequency. So when we did the same thing for the ZPEG promoter, so a new gene drive using this better promoter, we saw that the gene drive was able to spread up to almost 100% frequency in the cage. In fact, it got into almost every single individual except for about one or two. And those one or two that were remaining were unfortunate in our eyes enough to carry a resistant mutation. So we don't have zero production of resistant alleles anymore, because you still get them being developed in a normal male and female. You just don't get them developed from deposition. But by reducing the rate at which these mutations are generated, our gene drive was able to spread all the way up to almost 100% before it crashed and failed. And what we saw is that it resulted in a massive population suppression of around 99%. So we counted the number of eggs produced by this cage each generation, and it went from around 15,000 on day one down to around a few hundred at the peak of the gene drive. But at that point, there was such a strong selection pressure that those few mosquitoes that contained a resistant mutation came under very, very strong selection. And that's exactly what you see when we looked at the different mutations being generated. So when the mosquitoes are fit, resistance has very little benefit. Resistant alleles did not get selected while that gene drive was spreading, because while it was spreading, it wasn't causing an impact on that population. If you remember back to the beginning, the gene drive only causes a suppression once there are many homozygous mosquitoes in the population. At the point that it's spreading, when most of them are heterozygous, there's no real impact of having a gene drive. So the selective pressure is delayed and your gene drive can spread very effectively. And it's only towards the end that there's a big selection for any type of resistance. So we almost crashed them, but we didn't. So how can we prevent the generation of these resistant mutations? And what I mean again is these ones that are truly resistant because they encode a functional version of that essential gene. So we thought that maybe looking at genes that show some sort of conservation in nature might be the best way to find something that is unable to tolerate new mutations very easily. So if you were to look at the mosquito genome and you were to scan the whole thing, you'll find that there is a mutation once in every 2.2 bases. So in the wild, in Africa, people have gone and sampled throughout different countries the mosquitoes, and they've sequenced the entire genome, and they've done this for a few thousand now. And what we find is every 2.2 bases, you can find a difference between one mosquito and another. And a gene-drive target, so a CRISPR guide RNA, will recognize a sequence that is about 23 base pairs long. So if you think about this, that means that on average, there will be at least some mosquitoes that are naturally resistant to any guide RNA we were ever to release into nature. So even if we were to suppress populations in a laboratory setting, we have this massive problem that once we get to the field, pretty much everything is going to be resistant already. So we abandoned gene drives, and now I'm going to change my talk and talk about something completely different. I'm going to talk about the insect sex termination pathway. So mosquitoes, when they're choosing whether to become a male or female, they have an initial chromosomic determining signal, and that's very different between different insect species. So it could be whether you have a Y chromosome, it could be the number of X chromosomes, or it could be the presence of a single gene, which may be on even an autosome. But this initial signal activates a master switch, which activates something called the transformer complex, which then activates these two genes called double sex and fruitless. And these two genes are present in every single insect species you can look at. But the master switch and the original signal that tells you whether you're going to become a male or female can be very, very different between different populations of insects. And this double sex gene is actually conserved in all insects. And if you were to look at the structure of that gene, it's very, very similar. So the proteins, although they can be different, they have a very, very similar structure in different insect species, in particular, one region. And that region is a female-specific region. So when I talk about the double sex gene, how does it tell a mosquito or any other insect whether to become a male or female? It does that by differential splicing. So it gets spliced into either a male form or a female form. So below, we have these red lines, and that shows you the different exons that are included in the female form. And above, we have this male form. And the difference is just one exon. It's this exon 5. And exon 5 is entirely specific to females, and you don't find it in males. And it turns out that if you look at the DNA sequence of this exon 5, it is almost exactly the same in all mosquito species that you look at. So this is interesting. And then led us to think maybe we shouldn't abandon the idea of gene drive, that maybe we actually can find something which is under some evolutionary pressure to remain the same. So I told you that every 2.2 bases is variable within a species. So if you find a stretch of DNA which is the same in different species, this is very surprising. And when you see that it's the same in a whole range of different species, there's something more happening. There's some reason why this region is ultra conserved. And this piece of DNA is perhaps the most ultra conserved region of mosquito DNA in the entire genome. So we thought, what happens if you were to mutate it? So we introduced a GFP inside and we mutated the exon 5. And in these pictures, you'll see a male mosquito with his fluffy antennae, and he has these claspers at the back that he uses to capture females and mate with them. And the female, she has these thinner antennae. She has the proboscis that's capable of biting people, and she has no claspers at the bottom. But when we mutate this exon 5, what happens is you end up with these pseudomales or intersex mosquitoes, and they contain, they sort of carry features that are half male and half female. And the result is that they're unable to bite, they're unable to reproduce, and they're unable to transmit malaria. So previously, I showed you we targeted this gene needed for producing eggs, and this was this gene ARGUP007280. And when we made a gene drive, it spread for a few generations, but then it failed because of these resistant mutations. Well, I can tell you that we then switched our strategy to target the double sex gene, specifically this ultra-conserved region in exon 5. And our gene drive this time spread from around 20% to 100% in around 11 generations. So this is the first time that any gene drive had been shown to spread to an entire population without failing from resistance. But think about what it does. It's going to convert females from being normal to being intersex, and the intersex are unable to bite or reproduce. So when we counted the number of eggs generated each generation, we saw that it went down from around 30,000 eggs on day 1 to 0 eggs, and these populations were completely eliminated. So as the gene drive reaches 100%, there are no more normal females left in the population, and those entire populations crashed. So it was the first time that gene drive has been shown to spread to an entire population, and the first time that gene drive technology has been shown to suppress and eliminate populations in a small laboratory setting. So I'll talk a little bit about off-targets. So one of the questions that people have is, could these guide RNAs be cutting somewhere else in the genome and generating mutations that we don't understand that could perhaps make a mosquito better at transmitting malaria for some reason or another? And there's a technique where you can take the entire genome of any species you want, and you can cut it up and turn it into these small circles, and then present it with a single guide RNA and Cas9. And you can measure what gets cut and what doesn't through sequencing. So it's an unbiased approach to work out any regions of the genome that might be susceptible to a guide RNA. And you can do this for mixed populations of genomic DNA from wild-caught mosquitoes. So we did this for the actual double-sex target site, this ultra-conserved region. And what you find is that it cuts an awful lot at the original site that we were expecting. So this is an unbiased approach. So we don't tell it where it's going to cut. We just put the guide RNA and Cas9 in, you sequence whatever got cut, and what comes out as the top thing is the original target site. So that's exactly what you would expect. And what we have is a very small number of reads for these other sequences which do not resemble the double-sex target site very much. And this number of reads is below the threshold we would usually use. So it looks like there's no activity. There's no activity at any potential off-target through this circle-seq analysis. But maybe that means our analysis isn't very good, or maybe it means that we're unable to detect these types of mutations. So what we did was we went back to that original gene drive, targeting the gene 7280, and we performed the same circle-seq analysis, and we searched for those mutations in our mosquitoes. And we found that we did find a significant number of reads for several potential off-targets. And when we looked at those off-targets in our mosquito populations exposed to the gene drive, we find mutations. So we do have some evidence of off-target activity, but it's very low-level activity, and it's only for this this gene drive with this poorly designed 7280 guide RNA. So we expanded our analysis to look at a whole range of other sites in double-sex, sites that usually you would never consider looking at based on circle-seq. But because we didn't find any mutations for the first main potential targets, we looked at anything that was even 4 base pairs different from the intended target site. And we scanned through 130 potential off-targets in the mosquito genome, and we found that none of these were being cut by our double-sex gene drive. So it seems that we have no evidence for any off-target activity, whether at sites that we would have predicted using bioinformatics or at sites that we find using this circle-seq unbiased method. And in fact, we also tried to see if we could force mutations to happen by adding this deposition that I talked about earlier. So we know that deposited Cas9 generates far more mutations than using the upgraded ZPG promoter. So we crossed our gene drive mosquitoes to a strain expressing this Cas9 to try and force mutations to happen at any potential off-target. And again, we found no evidence for any mutation happening at any off-targets. What you see here with this little blue in the middle, this is actually a rare polymorphism that was in our wild-type colony, and through a bottleneck became very abundant in our 2 populations that had the gene drive. But we confirmed that these mutations were not generated by CRISPR. There's something that was already circulating in our colony, and through whatever selection process we used when creating our original gene drive, there's something that our gene drive was created in. So what next? So in order to get a gene drive to the field, you need to go from laboratory studies into large indoor cage studies, potentially large outdoor cage studies, and then small scale isolated releases. But when we talk about a gene drive, because a gene drive is something that can spread and something that is unlikely to respect national borders, it's something that we need to be very sure about before we ever get to the field. And because of that, these intermediate steps of large indoor cages and large outdoor cages are absolutely essential to the development of a gene drive. And it's not obvious that a technology that works in a small cage in a lab setting in London is going to work in a large cage facility. And the reason for that is that when we modify any organism, you often introduce a fitness cost. And those fitness costs might be subtle. They might be a cost in how able they are to compete and mate. It might be a cost in how able they are to take a sufficient blood meal or how many eggs they lay. And in a small cage setting, these things are largely irrelevant. Unless the fitness cost is incredibly high, a gene drive is likely to spread even with a substantial cost. But in large cages, you can have a more natural setting where most mosquitoes will be dying anyway, simply because of the conditions that we keep them in, something more natural. And because of that, you have a much greater selective pressure for any fitness advantage. And so a fitness disadvantage that could be caused by a gene drive could be detrimental. And in fact, non-gene drive technologies have failed when transitioned from a small cage to a large cage setting because of fitness costs that were not obvious. So the things that we specifically were interested in is we wanted overlapping generations. So for this reason, if your gene drive is very effective for the first blood meal and then they die, you might find that that works very well in a small cage where you have discrete generations of populations. But when you allow them to sit there in a natural environment, none of them survive long enough for the gene drive to spread. And we also wanted to look at swarming behavior, competition for mates, and active searching for food. And what we found is that when we release our mosquitoes into this environment, the gene drive spread very effectively, but it's very noisy. So on the bottom, you see these two different release scenarios, one with a slightly higher release scenario and one with a slightly lower release scenario. And both of them spread within the course of around 1 year. And over that course of 1 year, the spread is very, very stochastic, but it does go up. And once it reaches 100% frequency, those populations crashed. So on the top, you see the number of eggs produced, and we have anywhere from 1,000 to around 20,000 eggs being produced in any one generation. And then that very rapidly crashes as the population is entirely modified. And here I've just got a picture of what that looks like. So we have these 6 different cages. We have little areas for the mosquitoes to rest. And we have a simulation that tries to get across the sunset and the sunrise. And that actually manages to get the mosquitoes to engage in swarming behavior, which is something that you can't do in a small cage setting. So I wanted just to get on to one last thing, which for me is one of the most interesting parts of this entire talk. It's what might happen if you were to take a gene drive and release it in Africa. And so we have facilities in Burkina Faso, and we've been doing modeling for what would happen if we were to do trials in and around the region in Burkina Faso. So on the left here, you see a map which shows in the middle Burkina Faso in yellow, and it shows around it Mali, Niger, Benin, Nigeria, Togo, Ghana, and Cote d'Ivoire. And what I'm going to show you is what would happen if you were to release your gene drive at several locations just a couple of times. And what we're going to see is that if the populations are red, then that means that there are well over, 100,000 mosquitoes in that population. And if the population is orange, that means it's around 10,000 to 50,000. And what you'll see is that populations are living, breathing things. And that between wet and dry seasons, you'll see a natural flux from very high numbers of mosquitoes in any one population, any one circle, to relatively low numbers. And that will go up and down naturally. But after a gene drive release, you'd expect to see it spread and start suppressing those populations. And I want you to imagine what you think might happen. Are we going to release our gene drive and see it spread just outwards from one position, like a big wave coming out? Are we going to see the gene drive spread for a while and then fail? Are we going to see complete eradication of the mosquitoes wherever the gene drive has spread? And how long does that take? And I think it's quite interesting to think about these things because it's not always too intuitive. So once these circles start getting invaded by the gene drive, what we might see is that they convert going from this red and orange, which is high population numbers, down to the blues, which is low population numbers, and potentially elimination, which would be this darker blue. So we release our gene drive, and initially what you see is that, sorry, we haven't released the gene drive. Let me go back. So for the first few years, we're just seeing the natural flux from wet and dry seasons. But when we release it now, what you see is that very, very rapidly, the gene drive spreads almost everywhere. So it doesn't spread outwards in one wave from a central point. What you see is that because of the migration of mosquitoes, it spreads very, very rapidly through an entire region and causes suppression, which is very drastic. But what you don't get is you don't get population elimination. Throughout this entire process, even after release, the mosquito population in Burkina Faso is still going up and down during the wet and dry season, but the total number is now drastically reduced by around 95%. So even with our most powerful gene drive that spreads incredibly well and doesn't generate resistance, these are all the assumptions, which may not be true, but the assumptions that we use for our model. Even in that optimal scenario, you do not get eradication of the mosquito. You get a strong and sustained suppression that is an average of 95% over this 4-year period. And we find that it lasts much longer than 4 years. And that's an average suppression. Obviously, wet and dry seasons, there is variation. And we do have one more trick. I'll say it quickly in 2 minutes. Just because we didn't see gene drive in our initial experiment, didn't see resistance in our initial experiments, it does not mean that resistance is impossible. So we've been searching around to see if there are other target sites that we could target using guide RNAs in a multiplex gene drive. And this is that region of double sex exon 5, and the original target site we go for, this T1, this is the one we've been using, but we've identified several other target sites which seem to show a similar or even greater level of nucleotide conservation. And the idea is that if you target one site, then any resistance at that site will come under a strong selection and your gene drive will fail. But if you target many sites, then resistance at one site doesn't matter. And that's because you can cut the other site and the gene drive still works. Now, something which I've failed to get across very well in my slides, but I'll try and explain in words, is a concept which sometimes is difficult to understand. So if you have resistance at one site, but you have a multiplex gene drive, you might say to me, well, you get resistance at one site, and that takes an amount of time. Let's say it takes 1 year. And then it's just going to be an amount of time before you get resistance at the second site, and then an amount of time before you get resistance at the third site, and then your gene drive will fail. But I would argue that that's not actually the case. If you have resistance at, let's say, site number 3, like we have in this picture here, because sites number 1 and 2 are not resistant, they're going to get cut. And your gene drive is going to replace that entire locus with a gene drive. And in doing so, it removes the resistance at site 3. So every time you get resistance at one site, but not the other, your gene drive will remove that resistance from the population and replace it with a gene drive. So in order to have resistance, you actually need resistance at all 3 sites in the same mosquito at the same time. And because of that, you can reach a statistical chance, which is incredibly low enough that a gene drive could be effective for several years, to the point that we can reach a real suppression and make a real difference in malaria transmission. And we've built these gene drives and they work. And surprisingly, they even seem to be fitter than the original single guide RNA versions. I don't want to go too much into detail with that because I notice I'm spending a bit too long talking. And so in terms of transition to the field, we have a number of steps. We need to work within local, national, international guidelines and regulations. We need to do phase testing that needs to start in laboratories in small cages. It needs to move towards large cages. We need to use modeling. Modeling is going to be an essential component of this process at every step of the way. And we need to work with community and stakeholders. We need to engage early, and something that we've been doing even before CRISPR existed, we were working with stakeholders and working with the communities to say, if we build this technology, what are your concerns? And how do we take that into account at the point of development? We have ethics advisory committees, which include a number of representatives from different religious groups, and we work with key influencers. And as part of this project, we're building capacity in Africa, we're training the next generation of scientists, and we're working within existing control programs. And finally, everything that we do, of course, is going to have to involve rigorous safety studies before release, independent risk assessment, and addressing ecological concerns. So for current vector control, the most effective method is to target the mosquito. And this is something that gene drives do as well. There's something that is entirely in line with what we already do to control malaria. Gene drives can work very, very fast because of this mechanism of spread. They can be very, very cheap because the mosquitoes are the thing that do the work. They're the thing that spreads it. So it's not very resource intensive as compared to widespread use of insecticides. And mosquitoes, because they are the thing that spread, they can bring that intervention into the places that need it the most. And finally, I would say, because gene drives are species specific, you can actually impact those few species that transmit malaria without affecting the 3,500 mosquito species that do not transmit human disease. And I'll give a quick thanks to everybody. This is part of a very large project that involves more than 180 people across a number of different countries in Europe, North America, and sub-Saharan Africa. And we have groups that are very focused on building those resources and technologies and infrastructure in Burkina Faso, Mali, Uganda, and Ghana. And we have most advanced in Burkina Faso, where we've already released non-drive mosquitoes to start paving the way for an ultimate gene drive release in the future. So apologies for talking a little bit too long, but special thanks to everyone, and I'll open it up to some questions.
All right, thanks for your talk. Yeah, we started a bit late to had some introductory words in the beginning. So, I mean, lots of people were here at the beginning stage, so I guess everyone was interested. And we already had a question by Greta. So, Greta, you can turn on your microphone now and ask your questions. And meanwhile, people can keep asking questions on the Q&R.
I think it was answered already later that it was not really released ever, because my question was if there are cases where the mosquitoes became resistant and it had a negative effect on the population of a region and the cases of malaria then have risen. But I think it was explained later that it was just studied in laboratories and has not been yet released to the field, really.
So that's right, but I think that there's something interesting in your question, which is what might the impact of resistance be after a gene drive release? And if you have a situation where 4 or 5 years after a release, where you have a widespread suppression and malaria transmission goes down to very low levels, you could have a situation where malaria starts to come back if the gene drive technology fails. And we know that one of the reasons why malaria deaths are not higher than they are right now is because of natural immunity in regions where you have high malaria endemicity. And so there is a real risk that if you have a technology failure after a few years of success, that could actually be a real problem for malaria control. And so it's not so simple to say, oh, it worked for a while and now we just need to develop something new. You actually need to be very serious about what is a long-term strategy and how do you mitigate the effect of a resurgence of malaria in an area where it might have disappeared for a few years and you might have waning immunity.
Happy with the answer?
Yeah, thank you.
Okay. I think Nicolas might have a question too.
Yeah, so thank you. That was very interesting. I was very curious about the different in the mutation spectrum, or the types of mutation between males and females. And so I was wondering, yeah, is it because the mutation spectrum is different? So the way to repair DNA is different in general in mosquito, or in this species? Or is it because the promoter, for example, is Cas9 is expressed at different time in the cycle, for example? So maybe if it's expressed earlier in the females, then all the progenitor cells, you have the cut in the progenitor cells. Whereas if it's later in the males, then you would see the pattern you would expect the pattern you see. And so I was wondering about what's your opinion on this, and if you, I don't know if there are mutation accumulation lines in mosquito where we know the mutation spectrum. So maybe if you could comment on this difference between males and females.
Yeah, so I don't have all the answers to that, but I do have some ideas of what's probably going on. And you're quite right that clearly what's happening is that in the females you have an early event, and that gets into a larger fraction of those germline cells that come from that event. Probably it's also related to the fact that in females this is all done in batches. So you have just a certain number of eggs that are developed in each gonotrophic cycle. And that's coming from one sort of string of progenitor cells. Whereas in the males, you have a very, very high number of sperm that get mixed in a bag, where these mutations could have happened at different time points in that development. I'm not so sure if it's related more to the timing of the expression of the Cas9 and how early that is, or related to the timing of the DNA repair, and that being a natural difference between males and females. I have a feeling it might be the DNA repair, which means that it's something that is probably practically impossible to sort of get around that in males. So I think that reducing the diversity of mutations and reducing the extent, the gain of mutations is important, but you can't rely on that. I don't think anyone's ever going to reach a point where you have 0 mutations being generated. So you need to combine these different approaches. But yeah.
Cool, thanks.
So yeah, there was lots of information in your talk, so I could have several questions, but perhaps a question a bit naive from which perhaps is for the case for many people in the audience like me, is that this, what you presented in the end with this construct with 3 target sites together, does it have to target the same gene, or does this construct, can they work if they target different genes?
Yeah, so
and how does this differ from having a larger, just one target site that's more or less big, basically?
Yeah, so let's see if I can just pull up that picture here. Okay, so here we have 3 different target sites that are all within the same gene. And with a multiplex gene drive that spreads in the way that, I mean, one of the complicated things is that gene drive is becoming such a crazy field now that there are so many different ways of building these things and tweaking it that it becomes difficult to talk about one thing. But I think when most people think about gene drive, they think about something the way that I presented it today. This sort of, you cut something and you copy it inside, and that's it. This is like the most basic gene drive. And in this scenario, all of your target sites would need to be in the same region. So
yeah, I guess what I was saying would be multiple independent drives. That work better than this, or
yeah. So I would say multiple independent drives is something that everybody is interested in, and that would, like, when we first developed this idea, the expectation was that a single gene drive will fail. And what you would need is that you would need several different gene drives targeting different genes at the same, like, one gene drive targets one gene drive targets a different gene, and you release several of them at the same time. And then you have a follow-up where 2 years later, you release the next few gene drives. And then we found this sort of one major problem and then one major bit of luck. And the major problem was that resistance is probably a bigger issue than we thought. in the sense that many of these single guide gene drives just wouldn't be able to spread in natural population. You just look at the diversity and they will never work. From day one, they just won't work. So all of these, like our original designs, they would just never make it to the field, not the way we're anticipating. And then we found this target site in double sex, which is so good that a single gene drive might actually be enough if it's multiplexed. So if we could find more sites like that double sex target site, more ultra-conserved sites, that would be the best possible situation.
Yeah, instead of having multiple that spread halfway and sort of relay each other or something like this.
Yeah, I would say each gene drive needs to be effective on its own. So it absolutely needs to be effective on its own.
Okay,
but you would want to have multiple different gene drives at the same time, and that would have an effect of mitigating resistance. But the way it mitigates it is a little bit different. So it's not that it stops those mutations from being generated. It's not that it stops resistance from happening. It's that it reduces the population in an independent way that makes it more difficult for those resistant mutations to become established.
All right. There's a question by someone else, Caroline Montagnani. So Caroline, you should be able to turn on your mic now.
Okay, do you hear me?
Yes.
Yes.
Yeah.
Sorry, my camera is not working very well. I have a kind of a naive question as well. It's about the field strategy. You mentioned that there's 3 species of mosquitoes that are transmitting malaria. Mm-hmm. So how do you, what's the plan? I mean, you're going to focus on one species, then on the other 2? Aren't you afraid that could be kind of an endless pursuit because you're going to have to crash all the species once at a time at the same time?
Okay, so we can actually look at what's already happened in traditional vector control to already inform on this question, I think. So there are regions where Anopheles gambia used to be the dominant malaria vector, and now you no longer find it. And instead, you find that the dominant species biting people is Anopheles arabiensis. And the reason for this is very interesting. It's actually because Anopheles gambia likes to bite people indoors at nighttime, and specifically people. Whereas some of these other malaria vectors, they are more generalist, so they might not focus on humans, and they also might not focus on biting indoors, and they might bite at different times. So the combination means that we developed malaria control that was targeting the species that is transmitting most malaria, and that was Anopheles gambia. So that's mosquitoes biting indoors at nighttime when humans are sleeping. But because we focused on those technologies, it didn't have such a great impact on the less efficient malaria vectors like Anopheles arabiensis and Anopheles funestus. So then you say, okay, Anopheles gambia is no longer a problem in those regions, but now the major vector is Anopheles arabiensis and Anopheles funestus. So aren't we back to square one? And I'd say we're not back to square one. Those vectors are nowhere near as good at transmitting malaria as Anopheles gambia. They're not such efficient vectors for several reasons. I mean, one, they don't bite humans as often. So the chance that they bite someone, pick up the parasite and transmit it to someone else is less. And also the parasite is less well adapted to those species. And I would say it's not, there's often a concern that the parasites can become better adapted to different mosquito species, and that's true. But some of this happens on an evolutionary timescale, which is probably would take too long compared to the timescale in which a gene drive would work. And the other side of it is that even if they were to become very well adapted, for many of the mosquito species, they do not bite humans enough to keep malaria transmission going. So there are another 30, 40 mosquito species which can certainly transmit malaria, but malaria would be eradicated if you only had them in the world, because they don't transmit enough malaria and they don't bite humans enough to keep that cycle going. So we think that really, if you were to go for these 3, that would probably eradicate malaria from, probably the world, but large regions for sure.
Okay, thank you.
Okay, with the exception of other types of malaria, Anopheles stephensi and things that I realized that I'm sort of got a little bit wrong there. But for sub-Saharan Africa, those 3 species would be the biggest, most major suppression of malaria.
Okay,
Yeah,
great.
thank you.
Anyone from Mivejek wanted to react on malaria and mosquitoes? No, we don't get a reaction, so it must have been correct what you. Okay, enough jokes.
I almost said something a bit wrong there, but
well, they didn't notice
thought
at least. No one is reacting. Okay, so I think we're running, I think, a bit out of time. So thanks a lot for your talk and the questions. So and the responses to questions. Sorry. So this video will be on YouTube shortly. And I want to tell everyone that next week we should have our first in-person seminar, hopefully for a long time. The place will be announced shortly, probably at the university. Probably. So thanks again, and see you next week at 11:30. Bye-bye.